Hello everyone, my name is Sara Khan from group 322. My topics are conformations of polypeptide chain and hydrogen bond. So, conformation of polypeptide chain. For understanding protein structures, conformation of polypeptide chain are necessary. The most stable conformation, total energy and intermolecular hydrogen bond energy should be minimized. Pauling and Corey have found most stable conformations on the basis of X-ray data, considering light packing of chain with maximum number of hydrogen bonds. So, various properties of peptides and proteins depend not only on their component amino acid and bonding sequence but also on the way peptide chains are stretched, coiled, and folded. Conformational segments are described by dihedral angles. So, here are the five factors that influence conformational equilibria of peptide chains. First one is planetary of peptide bonds. Second one is conformation are defined by dihedral angles. Third, steric crowding and neighboring groups. Fourth, repulsion and attraction of charged groups. Fifth, hydrophilic and hydrophobic character of subsequent groups. So, according to these factors, polypeptide chain can be folded into alpha helix, beta sheets, turns, and loops. So, now coming to alpha helix. It is proposed in 1951. Dimensions are steric constraints and opportunities for stabilization by formation of hydrogen bonds. Features of alpha helix. A. Relative location of the donor and acceptor atoms of hydrogen bond. B. Number of amino acid units per helical turn. C. Distance that turn occupies along the helical axis. So the perfect alpha helix have conformation with minus 57 degree and minus 47 degree, and each helical turn includes 36 amino acid residues. Distance covered by turn is 5.4 Armstrong, known as pitch, but for natural proteins it is very different. Now, alpha helix can be right or left-handed screw. Right-handed helix are energetically more favorable because there is less steric clash between side chains and backbone. All alpha helix found in proteins are right-handed. So here you can see hydrogen bonding pattern in alpha helix, how it is arranged. It is given here. So now coming to the stability of alpha helix. Hydrogen bonds are formed between hydrogen attached to electronegative nitrogen atom of peptide linkage and electronegative carbonyl oxygen atom of the fourth amino acid on amino terminal side of peptide bond. All hydrogen bonds together provide stability to alpha helix. So as I have mentioned before on the before page that left-handed helix are very rare and right-handed helix are very common in alpha helix. So now coming to beta sheets. Beta conformations is an extended form of polypeptide, the backbone of the zigzag rather than helical structures. Zigzag polypeptide chains can be arranged side by side to form a structure resembling a series of pleats called beta sheets. Beta sheets are composed of two or more polypeptide chains called beta strands. Beta sheet is formed by hydrogen bonds between protein strands rather than within a strand. The structure is stabilized by hydrogen bond. Okay. So now next coming to beta turns and loops. Beta turn is a region of the protein involving four consecutive residues where the polypeptide chain folds back on itself by 180 degree. Most proteins have compact folded structure and nearly one-third of the amino acid residues are in turns or loops where polypeptide chain reverse directions. These are connecting elements that link successive runs of alpha helix and beta strands. Most common structure element is beta turn or hairpin turn or reverse turn. Most elaborate structure comprises the loop. Okay. So now next my topic is hydrogen bond and structure of water. So we know that water is a very important component, a very essential component in our life. So all living organisms are dependent on water. The structure of water is the basis of its unique properties. It have very unique properties, and it is really very essential. The most important property of water is the ability to form hydrogen bonds. So within a hydrogen molecule, the bond is between the oxygen and hydrogen, and they are very highly polar. Okay, so that oxygen is partially negative and hydrogen is partially positive. So hydrogen bond are weak attraction between partially negative oxygen or one molecule of water and the partially positive hydrogen of diffusion of a H2O molecule. Hydrogen bonds can form between H2O molecules or between water, another charged molecule. So now, secondary structure of protein chains are stabilized by hydrogen bonding. Hydrogen bonds also play a key role in conformation structure of nucleic acid and carbohydrates. And biopolymers function in aqueous environment. Special properties of water which make it an indispensable component of cell and organism are also determined by hydrogen bonds. Presence of hydrogen bonds extracts a strong effect on the physical and physicochemical properties of substances. Intermolecular hydrogen bonds determine the association of molecules. Associated substances are characterized by relatively high heat of vaporization, high melting and boiling points, and by large differences in melting and boiling points. So now coming to hydrogen bond. Hydrogen bond are weak attraction between partially negative oxygen of one H2 molecule and partially positive